Hello everybody, biomedical lab science student and laboratory technician, Juha here. Today I would like to show you something that's very near and dear to me. My specialty, so to speak, and love in this whole biomedical lab science field is microbiology. So today I will be showing you pharyngeal secretion, inoculation, and spreading on a real sheep blood agar plate. And this is actually quite a rudimentary thing to do as an MLS. I'll grab all the prerequisite materials and I will come back to you. Thank you. So yes, this work consists of actually inoculating the blood agar plate. This is done with a patient's pharyngeal secretion sample, which is usually on a kind of a Q-tip sort of a material, which is submerged in agar jelly as well. But in this video, I will only be showing you a facsimile of that. It is just a normal sterile cotton swab. And after that, the inoculation part, we will be spreading the sample on the plate using these inoculation loops. These are 10 microliter inoculation loops. This is very like a commonplace item found in microbiology labs everywhere. So thank you. Okay, welcome back everybody. So here we are at the workbench in the microbiology laboratory, and the first thing we do is actually mark the plate medium according to the sample we're actually going to inoculate it on. So I've taken the liberty to already do that, so I've marked it as PS BACTV. So that's pharyngeal secretion, and it's for bacterial inoculation, and the date is visible here as well, the 4th of November 2025. And what we can also mark here on the plate is actually the type of the medium used on this particular plate. This is just a non-selective sheep's agar plate, so basically what that means that everything is going to grow on it. The bacterial type or morphology doesn't really matter here, so everything will grow on this. And normally we would mark it, but usually these commercially produced plates have the type already etched on the plate, as it says right here. So it's sheep's agar, non-selective. Okay, so here we are with the patient's pharyngeal swab sample. And what we do is we open the plate, flip it over, and then we very, very gently spread it here, without hitting the edges, preferably. We use approximately 1/3 of the circumference of the plate for the spreading. Then we discard the sample stick. We take the inoculation loop and we do a spread here for the sample as well. So the idea is to, as gently as possible, move the sample from the original part to the next swab location, then turn the plate, then do the same thing for the secondary sample location, into the tertiary sample location, and then we rotate it one more time and rinse and repeat. And hopefully now at this location, this final location here, we will have single colonies of the possible pathogen that's growing in our patient sample. And then we, of course, discard the loop as well. And then we flip the plate back down and we're done. And here we are with the final product, so to speak. So this is the completely inoculated plate that contains whatever was growing in our patient's throat. So the pharyngeal secretion sample we just inoculated on this plate and spread it. So now the next part would be to immediately take this and put it into the incubator. And as a clinical sample, clinical samples are usually inoculated at body temperature. So in this case, of course, 37 degrees centigrade. So we would set the incubator for that specific temperature, and we would leave this plate there to incubate for approximately 24 to 48 hours. And if you made it this far into the video, I would like to thank you for your interest in this subject matter, and I will see you in the lab. Thank you.